So section 3, we're going to talk about the adaptive immune response, which is our specific defense system and our body's third line of defense. If our adaptive immune response fails, then we end up with some sort of disease or disorder, or even potentially cancer. So specificity is a benefit of the adaptive immune system. One negative attribute would be that it takes time to make these cells that are specific to a foreign antigen. So the priming would be a negative or a shortcoming. General characteristics, specificity, which we already talked about. Another one would be that it is systemic. So it's not restricted to the initial site like we saw with the second line of defense. Another benefit is that it has memory. So if we were exposed to the same antigen on a second exposure, or potentially even a third or fourth, we can mount a much faster and more robust immune response against that particular antigen. There are two branches of the adaptive system. One is going to be humoral immunity, which is antibody mediated. The other one's going to be cellular immunity, which is cell mediated. The big difference between these two branches of immunity are going to be the lymphocytes involved and the targets that they're after. So with humoral immunity, we are working with B lymphocytes. B lymphocytes are going to differentiate into plasma cells, which are just cells that are going to pump out huge amounts of antibodies that eventually are going to be released into body fluids. There is a misconception that antibodies will actually go in and destroy foreign antigens and microbes. That's not the case. All antibodies do is go in and bind to the foreign antigen, basically inactivating it and then marking it for destruction by another type of immune cell like phagocytes or complement or something like that. Because antibodies cannot cross the plasma membrane of cells, they are limited to the antigens that they can bind. So they have to be on the surface of cells or pathogens, and they have to be extracellular targets. When we're talking about cellular immunity, we are dealing with T lymphocytes. T lymphocytes are able to go in and identify things intracellularly or within the cell. So cells that are infected with viruses, even bacteria and parasites, and sometimes even cancer. And they can kill either directly by killing the cell itself or indirectly by activating the inflammatory response or other cells like macrophages. So we've mentioned antigens a couple of times, and now we're going to talk a little bit more about what exactly is an antigen. So an antigen is anything that can mount an immune response. So we have on our own cells, we have antigens on the surface of all of our cells that we refer to as self-antigens. And then pathogens and debris have antigens on their surface that we would want to hopefully recognize as being foreign. So it's actually a contraction of two different words. So antigen is a contraction of antibody generating. So it's actually foreign antigens that are going to cause our B lymphocytes to proliferate into plasma cells to produce antibodies against them. So we can have either a complete antigen or we can have a small piece of an antigen, in which case we refer to it as a hapten. And each antigen has its own little signature thing sticking off of its surface that we refer to as antigenic determinants. So like I mentioned, we have a whole antigen that can be recognized as being foreign. These are ones that are capable of what we call immunogenicity, meaning the capability of stimulating proliferation in specific lymphocytes, and also reactivity, where they're able to react with those activated lymphocytes and antibodies that were released by immunogenic reactions. So some examples of complete antigens include nucleic acids that are being injected into cells, say by viruses, lipids, polysaccharides, or sugars or carbohydrates on the surface, and even some foreign proteins. Our little pieces of antigens that we refer to as haptens, these are really, really small, so they're not considered immunogenic by themselves. They wouldn't mount an immune response. However, a lot of haptens, or these little pieces, will go in and bind to our own proteins in our body, which is not a good thing. Because if these haptens bind to our own proteins, then our immune system recognizes both the hapten and our own protein as being foreign. It can cause our immune system to mount an attack against it. So some examples of incomplete haptens or antigens would be poison ivy, animal dander detergents, and even some cosmetics. So this is showing you a little bit more about those antigenic determinants that I was talking about. Some antigenic determinants are more potent than others, and usually a single antigen can mobilize several different lymphocyte populations and antibodies of different kinds. So here's the antigen shown to you as the spherical shape in red. Its protrusions that are sticking off of it. These are the antigenic determinants. I probably should have used a different color other than red. But they're illustrated to you over here. So they're showing them and representing them as different shape to show that they are each unique to one another. So each antigenic determinant can stimulate the formation of a unique antibody. So because this antigen has 3 antigenic determinants, it can potentially bind 3 different antibodies. If we're talking about something that doesn't have a lot of antigenic determinants, that would be something like plastic. It's identical repeating units, so it has little to no immunogenicity. And this is why we use a lot of plastics to make artificial implants, so that we don't mount an immune response against it. But in general, the higher the number of antigenic determinant types we have on an antigen, the higher the immunogenicity and the stronger the immune response we're going to mount against it. So the last little thing in this section is we've mentioned antigens, antigenic determinants, and we talked about self-antigens, which are these little carbohydrate signatures that we have on the surface of our own body cells. However, we don't just have these little proteins or carbs sticking off the surface of our body cells by themselves. They're actually mounted on something else. And what they're mounted on is what's referred to as an MHC protein, or a major histocompatibility complex. And it contains a little groove where it's going to hold the antigen so that it can present it to different types of cells that we have. So if this is our cell right here, the MHC complex could potentially look like this thing right here that I'm drawing in. Say it looks like this U here. That would be the protein. And then inside of the MHC complex, there'd be a little groove where it's going to house the antigen, or basically present it on its surface.